Good morning, and thank you very much. I think this is the human part of the project where I get to actually share with you some of the patient stories and some of the issues that we've been struggling with. I also come to you this morning with pretty much of a heavy heart for so many reasons, because my work has been supported by NIH through the years, and in particular, the Secretary's Advisory Committee on Hereditary Diseases was dissolved yesterday. This is the program that was entirely responsible for bringing to the entire public the notion of detecting and treating rare diseases before they could happen. Excuse me. As I do with all my talks, though, I'd like to make you aware of the sources of my support over the years. Obviously, NIH and other foundations. Also, I advise several companies, and I have co-founded 2 companies, one of which I'm going to mention at the end of this talk. And this is the team that I work with. Everything I talk to you about is a group effort. We've come together to try to bring genomics into day-to-day care. Now, the way we got started with this was thinking about risk, cost, and benefit. Why are we not all using the genomics revolution more in our day-to-day care? Why isn't your doctor telling you to get your genome sequenced, your children's doctor telling them to get their genome sequenced in order to understand what you're at risk for? How is this going to get started with multi-omics if we can't get it started with genomics? So my career has been devoted to DNA first, how we bring DNA first into your life. And we've attacked that in 3 different ways. We were the first to bring a policy for secondary findings. If you get sequenced for a heart problem, should the laboratory look for a cancer predisposition? If so, how deeply should they look? How many genes should they look for? Who reports that back to you? That was the subject of literally hundreds of controversial papers until 2013, when I led a working group that developed the so-called ACMG, the American College of Medical Genetics and Genomics list, which arbitrarily to some, but we thought logically, put together 56 genes, which were the first 56 genes that anyone who gets sequenced should actually get returned to them. And I'm happy to say that that policy got adopted by the board. It's gotten promulgated to molecular labs all over the country and to some parts of the world. And now if you or your child gets a whole genome or a whole exome sequence for an indication, the laboratory will automatically look for, this 56 has grown to 81 genes that would be important for you and your family to know about. So that's the incidental or secondary findings controversy. The second area we've been very active has been the return of genomic research findings. It probably won't surprise you to know that throughout the world, if you count all GWAS studies and all sequencing studies, about 100 million people have had their genome done. 100 million. How many of those do you think have received medical feedback, medical information, medical utility from any of those genomes? How many? The answer is almost none. In the entire world, we have gone about sequencing people over and over for research, which is great, but we have not honored those participants by giving them anything useful back. So both in biobanks around the country, through policy statements in Global Alliance for Genomic Health, and specifically for US studies like the all-white Framingham study and the all-African-American Jackson Heart study, we have pioneered bringing genomic results back to research participants. It's not easy, and most research budgets don't want to budget for it, but we think it's a critical thing to do out of respect for people and out of giving them back something for their participation. And all this has set the stage for population screening. If you, I usually ask the audience, and if you don't mind, I'll ask you, how many of you have actually had your genome entirely sequenced? Okay, just a few of you. And this is a very sophisticated audience because many times people who've had a direct-to-consumer genetic test believe that they've had it. And of course, you know the difference because that's mostly array-based testing and we're talking about sequencing. What if there was an opportunity for every single one of you to have that information at a reasonable price and to use it in your medical care? And I think that's where we're going. And so many of the questions that I've been struggling with in my research are why is it taking so long? How can we overcome those obstacles? When we did that, the first group in the world to sequence healthy adults like yourself, we found something incredibly startling. We found that 15 to 20% of you are carrying a pathogenic or likely pathogenic variant from a monogenic disease risk. Now, obviously, you're not all going to get the disease. Maybe penetrance is only 50%. Maybe penetrance is 20%. But the fact that you're carrying this risk factor is something that you should know and your medical care providers should know. And when we look in a biobank, like even the Mass General Brigham Biobank, we look at people who are carrying these mutations who didn't know it, their doctors didn't know it. We find people with subclinical features of the disease in question and with misdiagnosed features of the disease in question. There is a lot more genetic disease out there. The whole notion that this is super rare has permeated our culture, but in fact, it's not. Some of the individual diseases are quite rare, but in the aggregate, they're incredibly common. If you don't believe the statistic, let me reassure you that Craig Venter's group published shortly after us and Mike Snyder's group published shortly after us and found exactly the same thing. It's just that people don't usually look in this kind of depth, and they certainly don't usually return information this kind of depth. Of course, we're all carrying recessive carrier traits, and many pregnancies are unplanned, but for those that are planned, we ought to know what the parents are carrying so that we can use advanced reproductive states to avoid catastrophic children. This group is very sophisticated about pharmacogenomics, and particularly in the Netherlands, people have demonstrated that on a population basis, this reduces costs and improves outcomes. And you know about polygenic risk scores. The world knows about polygenic risk scores. Academia is just discovering polygenic risk scores. But it does seem like there will be people at the tail end of these polygenic risk scores who have risk stratification, and it's very, very useful to know that. In the same manner, you might want to know if they're carrying a monogenic disease risk so that there can be added attention, added surveillance, and added aggressiveness to the treatments to keep them out of type 2 diabetes, heart disease, and common cancers. So there's a lot in the genome. Even before you get into multi-omics, even before you get into wearables, and before you get into big data and AI integrating, there's a lot that we could be doing. Now, this kind of is obvious when you think about it, but I found that many audiences don't realize that the more you look, the more you find. So it's better to do something than nothing. And so we've been helping healthcare centers all over the country try to integrate even just 10 genes for a few cancers, a few heritable cancers, and familial hypercholesterolemia. And even there, you can benefit 1% to 2% of the population. If you go with this ACMG list that I've been talking about, you can get up to 4% to 5% of the population. I have no connection with Invitae, but they have a health screening test of 163 genes. And you can see that there's an even higher population positivity there. And then, as I mentioned, in our research projects, we found a very, very high proportion. So it's expensive to do more genes, and the AI initiatives you've been hearing about at this meeting will reduce that cost. But for now, even getting some sort of panel off of the genomes will be hugely helpful. Now, I've been doing this for about 20 years, but over the last 10 or so, we turned our attention to the question of newborn babies. And newborn babies is a particularly useful group to consider sequencing. Number one, because they've got their whole life ahead of them, and you can kind of create this blueprint of risk. But number two, there are a lot of rare genetic disease that manifest in childhood. and more, they're treatable. As you've been hearing at this meeting and all the others you're going to, there is an explosion of cell and gene therapies, CRISPR, other forms of gene editing, enzyme replacement studies that are making these conditions treatable, but only if you find them in time. If you find them too late, the damage is done. So the logic of screening children early, something that's been talked about for 30, 40, 50 years, even before the Human Genome Project was online, people were imagining this notion of creating a blueprint of children. And I'm proud to say that in 2013, we wrote our first grant for this, and in 2015, we enrolled the first child in the world into the first systematic protocol, which we called the BabySeq or Baby Sequencing Project, a 4-day-old baby girl at Brigham and Women's Hospital. And along the way, we faced quite a bit of controversy at academic meetings. I would come to meetings like this and I would speak on this, and somebody in the front row would literally stand up and shout at us that this was unethical. There was going to be so much medical harm, so much psychological catastrophe, so much unnecessary medical spending that it was sure to be a damaging enterprise. We have now enrolled over 1,000 families, and we have addressed those controversies head on, and I'm going to show you a little bit of that data today. So the issue, of course, is that there's hundreds of conditions now treatable, whereas only 30 to 60 are actually looked for in conventional newborn screening. So the initial yield, when you look at a large number of genes, was 11%. And in our second cohort, that has continued and is now actually around 12%. And what we're finding, if you look hard, are a host of things that we did not expect. And they're not all, they are not all things that you would consider to be future-oriented. We actually found and continue to find that about a quarter of the babies we identify with mutations, if you look harder at them, you find that there are clinical features already there. And this we did not expect. We did not expect DNA screening to reveal ongoing disease. But for example, this baby with an elastin mutation already had narrowing of the baby's aorta and can be now followed by an echocardiogram. So this was pretty startling. We also were very concerned whether parents would be distressed. There was a great deal of literature that it would disrupt parent-child bonding. And so we carefully measured this with validated scales in a randomized clinical trial format and published in JAMA. And sure enough, parents who volunteered for this were entirely okay. Of course, they're not happy if they find their child has a risk variant, but they did not have catastrophic psychological distress. They were grateful for the information. And even in our project, where we kind of leaned into findings, whether they were conventionally treatable or not, parents were glad to have the information. They were glad to know about it. They wanted to look for early signs. They wanted to enhance surveillance. They wanted to be on the lookout for clinical trials. So obviously these are families that are future forward. They're volunteering for a brand new study. They're not necessarily representative, but still it was very reassuring that this was the case. We also are the first in the world there have been plenty of modeling exercises, but the first in the world to start to measure the downstream spending. And the takeaway from small sample size, but the takeaway from, again, our randomized trial of this is that there's so much noise in the spending of a baby from age 0 to 1 that extra spending for a consultation with a cardiologist or a consultation with a metabolic doctor after you find a mutation really doesn't show up too much. This does not count the spending that you did on the sequencing and interpretation, of course, which is its own cost issue. But even when you look at the kinds of follow-up and you exhaustively track, as we did, what each baby did in terms of seeing a specialist, following up, the differences between babies with monogenic disease risk and babies without monogenic disease risk was not that great. So it's not going to be a zero cost to society to do these things, but some of the catastrophizing about medical expenses was probably overblown. And now this has led to the question of, are results actionable? If I had to mention one question I get, every time I talk about this, especially to a family or to a group, it's like, yeah, but can you do anything about it? And like I've used the word treatable every time in every sentence, and then you still get that question. And I don't know what that's from. Is it from the history of genetics where we got started with Huntington disease and that was so obviously untreatable that people have gotten that stuck in their head? But yes, conditions that we find today are big picture actionable, and within that small picture, treatable. So what do those two terms mean? They're not really well defined, but I use them to mean treatable, meaning there's a specific treatment that either reduces the chance of the disease or limits the harm of the disease, limits the morbidity of the disease. Actionable means you actually can surveil, you can know it's coming, you can intercede sooner. So pediatric cancer is both treatable and actionable. Biotinidase deficiency is treatable. Even something like some forms of autism that you might detect with early genetic testing, there's now studies that demonstrate that if you enrich the environment early, you get a much better outcome for those children. So that's not treatable in the sense of a pharmaceutical, but it's certainly actionable. So we really struggled with how to define this. Turns out that the ClinGen consortium put together a semi-quantitative scale of actionability. Now, it's pretty crude, it's a little subjective, but it basically asks, what's the severity of the disease? What's the penetrance of the disease? And in case, there's some computational people out there who don't know that, that means the probability that if you carry the mutation, you're going to get the disease. What's the intervention efficacy, whatever that intervention is, whether it's a treatment or a surveillance, early detection, and then how tolerable is that intervention? Surgery is obviously less well tolerated than, let's say, a vitamin supplement. So in this kind of scenario, Huntington disease would be quite untreatable. You can see the shape of this diagram there, whereas Marfan syndrome would be quite actionable with a diamond shape. So in the first iteration of our BabySeq project, here are the shapes of all of the conditions that we found. And on that semi-quantitative scale, actually all of them cross their threshold for actionability, which is very reassuring that we're not finding a lot of information that would simply be troubling and that you can't do anything about. It's also interesting to think of penetrance in different ways. In the babies that these are all the babies that were positive for mutations in our first cohort. We've since done an additional cohorts, but in this first cohort, only 3 of the babies actually were discovered to have features of the underlying disease. They were surprised, and it was an exciting surprise, but it was still a surprise. Whereas, if you looked at the parent, most of these are dominant conditions, if you looked at the parent who was then traced back to be carrying the mutation, several of them were also manifesting clinical features of the disease, and all had been misdiagnosed as not having a genetic syndrome. Excuse me. So if you're thinking of just the infants at one point in time, the kind of combined penetrance is only 17%. But of course, penetrance takes on a new meaning when you're screening. You're thinking about how many of them will develop the condition over an indefinite amount of time. Maybe the 5 years, maybe all of childhood, maybe a lifetime. Same thing with the parents. And if you combine these together and think of the family unit as something that you're screening rather than the individual child, then you're actually coming up with a kind of cross-sectional penetrance of about 35%. That's a lot higher than I expected. That means that within a family, when you find these mutations, if this holds with a larger sample size, you are finding that 35% of the people that screen positive are going to be manifesting this, or their family members are going to be manifesting this. And That's pretty useful. I mean, we do so much screening in the practice of medicine for so much less yield. Again, it's as if we've all conspired, well, I'm talking about my own specialty, we've all conspired to minimize the potential benefit of genomic screening. And even more striking, we took the trouble to look for some adult onset cancers in children, something that's quite controversial because we have now robbed those children of the right not to know, true, but is consistent with the ACMG list that I designed back in 2013, where these are on the list to be disclosed if found in children. So we're violating one set of norms, but following another. And we were able to trace these mutations back by coincidence to the mother in each case, and none of those mothers, in fact, none of those entire families knew they were carrying this mutation. And I'm both proud and humbled to say that in the 5 to 6 years since we discovered this in these 3 babies, all 3 of those mothers, all 3, have had risk-reducing surgeries. If nothing else, the BabySeq Project may have saved the life of those 3 mothers, and you're going to hear from one of them in a minute. So what does this look like with real people? These are just a couple of low production value snippets from some of the parents. They aren't cherry-picked. Pretty much everybody in BabySeq had these same feelings.
We just thought, like, why isn't this done for everybody? I think part of the attraction of participating in the study was having that information and how empowered that information would be.
We just want our children to be that healthy and happy. And if we can know that, potentially the harmful to them, we can hopefully act.
When we got the results from my son, we had learned that he was a carrier of something called supravalvular aortic stenosis. You know, and I think about the football players that just fall over and die and keep on getting hit. That's what they do about it. Oh, no, they don't know anything some way we know. So I always kind of comfort myself by thinking, at least I know.
The most critical thing that they found with my son is that he has a BRCA2 mutation. I was told that I had a significant mutation, but it's a high propensity for a couple different types of cancers, and that I should start to look into that. I would rather my boys have me longer than we get something that I know is a high risk. Overall, just so happy that we have opted in and we know our risks that we're taking action steps to, just extend our life.
Let's be real. I mean, when you have a newborn, it's kind of like your whole world changes. So knowing a little bit of information is going to be helpful down there.
It was a really positive experience for our family, and we would participate again if we had the opportunity.
It's been a journey there, and we are happy that we have it.
I just felt more so empowered to have the information than not. And I think that, with everything going on in our world, we should know more so we can do our best work.
So after BabySeq came out as the first study and started publishing, other groups have joined forces and started to launch projects around the world, some with much larger sample sizes than ours. Perhaps the most interesting is the Generation study in the UK by Genomics England, which is going to aspire to enroll 100,000 babies and has been very, very thoughtful about bringing this into their NHS, where they have a single payer system. And of course, from a cost efficacy point of view, it makes the most sense to do this early and then amortize the cost and the benefit over the entire lifetime of a child, hopefully saving some funds along the way as you avoid some of the diseases that this early information provides you with. If you're interested in this, the organization I co-founded 4 years ago is called ICONS, International Consortium on Newborn Sequencing. It has now over 500 members, 50 from 50 different countries. It's been meeting in Boston, New York, London, has an upcoming meeting in London in October of this coming year. And frankly, one of the areas we're really missing is the computational and informatics input. So if there's anyone here who feels like they have a passion for improving this area, either because of your association with pharmaceuticals or a family member who has a rare disease, we could really use help. We could use a committee on AI and informatics in the icons, and I'd love to talk to you about that. It's a growing young organization. If you come to this meeting in London, there's just a palpable sense of hope and change that's very, very exciting. I'll finish up by saying that, as I mentioned at the beginning, I'm not sure what's going to happen to NIH research in our country, nor is our healthcare system particularly incentivized to offer screening and then allow the entire healthcare system to surveil and manage prevention. Genomics, I think, is the tip of the spear. You've been hearing so many sophisticated things here about multi-omics and integration of wearables and AI-driven discovery of pharmaceuticals. I hope I've convinced you that there is a lot of morbidity to be addressed here. I think you're aware that there's a tremendous acceleration in pharma attention to these so-called rare diseases, that they're molecularly driven, cystic fibrosis, sickle cell, spinal muscular atrophy, now have treatments that can radically cure or improve the outcome on these children. Dozens more are in the pipeline right now, hundreds after that. I heard one estimate that $1 trillion was being spent on molecular therapies for rare disease. Much of what I've heard here at this meeting is about the pharma side of this, but imagine if you could actually have a resource of 100,000, 500,000, 1 million infants whose genomes you actually knew, and you could look for them to populate your clinical trials and be there, be immediately available when a new treatment became available. So after a lifetime in academia, I've co-founded a startup company called Nurture Genomics. We are a very early startup. We'd love help from any investors in the audience. But the idea is to have an AI-powered clinical platform, not direct to consumer, but physician ordered and integrated with pediatric practice. And the idea would be to simply identify children with treatable genetic conditions and then navigate those families and those pediatricians to a knowledge base that would help them care for it. Because some of the instructions are that the pediatricians don't know what to do when they find these positive cases. It's not their fault. These are extremely rare. So there are some elements from home testing kits that can be used, but genetic counselor backed with telemedicine. AI can work in two important places here. As the last speaker mentioned, we need to more clearly automate the curation of variants, and we can do that in a dry lab format as sequencing itself becomes a commodity. And we need to bring AI into the knowledge base to continually update the care and treatment of 3,000, 4,000, 5,000 different rare diseases so that pediatricians have confidence in what to do next when they find this, and they can give the care to families. This needs to be integrated with medicine. It can't stand apart from medicine. So we've done a friends and family round on this. We haven't launched nationwide yet, but you can, if you have children or grandchildren, you can go to nurturegenomics.com and sign up on the wait list and be among the first to have this done for yourself. This is part of a vision for lifelong genomic medicine, whether it's through some sort of revolutionary change in our healthcare system, or whether it's through platforms. If our healthcare system in the foreseeable future is not going to help more with universal and lifelong care, somehow we still have to bring genomics and then multi-omics into lifelong care. And that's the dream that I think some of you have been pursuing your whole life. So here are some of the contact information for me and our research and social media. We'd be very appreciative if you reached out to us for either the academic side of things or the new startup company. There's our X links, Instagram, and my email. If there's anyone who has questions, I'll be here for a few minutes. I'll have to scoot off to the airport because I'm going to go talk about this at Main Stage TED next week. I'm terrified, but I'm hoping that by presenting some of this to the world, the shared mission that we all have to take some of these families and make life better for their children will be moved forward. And I appreciate all that you do, and thank you very much for asking me to speak. Is there time for any questions? We can take a couple of questions if somebody would like to come to the microphones.
Thank you so much for the talk. Very beautiful proof of concept work. I do have a question, which is generally in the US, as you already alluded to, regulation and insurance incentives are a big hurdle to getting new diagnostics into the clinic. Do you have any thoughts on how that can be overcome? We know in cancer, at least Foundation Medicine has managed to bring a couple of cancer diagnostics onto the market. So what are your thoughts on sort of doing that for babies?
Yeah, you're absolutely right. The insurance incentives are a barrier. I hope that there'll be a sufficient demand at some point. You know, the psychology's worked against genetic testing for a while, worries about privacy and discrimination. When people start realizing the benefits, I'm hoping that in the same way that they've sort of pressed forward NIPT, it's become part of employer-based care. There's more insurance reimbursement than there was, and that moved fairly quickly. I'm hoping this could move more quickly too. But you're right, it's a big barrier.
Thanks.
Yeah, hi.
Hi, Leonard Lipovich, Shenzhen Huayuan Biotechnology. More biological question. So it's been decades since the Genome Project was finished, and we all know very well that less than 1/3 of human genes encode proteins. Nevertheless, what I'm seeing at this meeting as a trend is that everything is unleashed upon the same good old protein coding genes in the BabySeq project. Are we taking any advantage of the fact that the vast majority of significant disease-associated variants are in non-coding regions of the genome? And how?
The short answer is no, we're not taking advantage of that. That is a layer of sophistication that has been beyond our first baby steps in this area. It is an important area, though.
Great presentation. My question is about epigenetics, because we know some of the diseases are also related to epigenetics. So I'm wondering, what is your take on this?
Sure. Well, combining genome sequencing with methylation, with proteomics, with metabolomics, even combining, as the previous speaker showed you, combining monogenic risks with polygenic risks are all going to refine these risks. And that's really important. But sometimes we're talking simply a level of awareness. If you are taking your child to a pediatrician, your child is not eating well, and they are stumbling on the stairs, and they are fatigued at night, those are the early symptoms of half of these genetic conditions. If you had a prior probability through a mutation that there's a cardiomyopathy or that there's a neuromuscular disease, you've got a lead. You don't need to refine that with epigenetics. You don't need to refine that with polygenics. You've got some sort of lead. I love all that refinement. I think we need it. But I think at a first measure, we're wasting the opportunity to establish prior probability and risk stratification for some very straightforward genetic risks.
Thank you.
Okay, thank you very much.
I have a question.
Yeah. Oh, okay.
Sorry for that, last minute. So my name is Mark Zheng. Question I have for you is, you share like a fact that I didn't know before. There's a large amount of sequence data available, and the fact is that a lot of information have not been returned back to the patient.
Yes.
So using that kind of same idea as you illustrate in the BabySeq project, how can we take advantage of already available sequence data, and what kind of issue we need to concern about if we do want to using the same approach? Because a lot of those publicly available data, I assume some is in private entity and some is publicly available.
That's
and
right.
some associate with certain identity of an individual, and most of them don't. So how can we take advantage of?
So it's a brilliant question. And the short answer is that so much of this genomic data is partitioned either in healthcare centers, nations, or specific research projects. So much of it was actually collected with consents that do not allow recontact, that do not allow sharing. So many of the nation level studies in the Middle East, Asia, and everywhere have do not want the data to go out of their borders. However, there are groups like the Global Alliance for Genomic Health that are trying very hard to pull variant encyclopedias together. And if you start from the get-go, like we're trying to do at Nurture Genomics, with very clear consent for reconsent and appropriate de-identified sharing with pharma and with the world, and you otherwise guard privacy very carefully, it's a difficult needle to thread. I think that you will find that there are biobanks, there are collections that will make themselves available to the world. So I think there's a group of hospitals that recently got together, right? And you guys must know about this, and decided to do a big genomics project and pull their genomics data together with their clinical data. That was kind of surprising to me because a lot of the genomics information in these hospitals hasn't been fully consented for that. But I'm glad people are trying to take chances with this, because I think the privacy risks are real, but they've been over-indexed on in comparison to the discovery benefits and the clinical opportunities. Again, thank you very much.